Hello everyone, welcome to Allied Bites. In this video, you are going to learn about introduction for the bacteriology and the bacterial taxonomy. Here I am referring Apurva Shastri book, that is Essential of Medical Microbiology by Apurva S. Shastri. What are the learning objectives? At the end of this session, the students will able to know important contributors in microbiology, understand the bacterial taxonomy, bacterial classification, and the nomenclature of a bacteria. Coming for medical microbiology. What do you mean by medical microbiology? Medical microbiology is the study of microorganisms and their role in human health and diseases. There are several branches under medical microbiology such as general microbiology, immunology, bacteriology, virology, mycology and parasitology. What do you mean by general microbiology? General microbiology is nothing but of study of general properties of microorganisms. Immunology is nothing but of study of immune system. Bacteriology, study of bacteria. Virology, study of viruses. Mycology, study of fungi. Parasitology, study of parasites. Then coming for the contributors in microbiology, here the first contributor is Anthony Phillips van Leeuwenhoek. There are many more contributors. Only I am going to explain about very few which are important contributors of microbiology. In that, the first scientist is Anthony Phillips van Leeuwenhoek. He was born in the year 1676. He was the first scientist to observe bacteria using single lens microscope. He was the first scientist who observed the bacteria in the microscope, the two single lens microscope. He named them as little amnaculus. Amnaculus is nothing but of animal. So he named them as little animals. Then the second contributor is Edward Jenner. He born in the year of 1796. He developed the first vaccine, okay, smallpox vaccine using the cowpox viruses. Using the cowpox virus, he produced a vaccine called as smallpox vaccine. So the name is given as vaccine. Then the third contributor is Louis Pasteur. He born in the year 1822 and he died in the year 1895. He is one of a professor of chemistry in France and he is known as father of microbiology. Coming for the contributors of Louis Pasteur, principles of fermentation, sterilization technique, pasteurization of milk, developed vaccine against anthrax, fowl, cholera and rabies, disproved theory of spontaneous generation, postulated germ theory of disease, first to use liquid media concept, and he is one of a founder of Pasteur Institute in the Paris. Then the fourth one is Joseph Leinster. He born in the year of 1867. He is the first to use diluted carbolic acid to sterilize the surgical instruments and clean the wound. He was the first scientist to use carbolic acid, that to diluted carbolic acid to sterilize the surgical instruments. So he is termed as father of antiseptic surgery. Then coming for the fifth contributor, that is Robert Koch. He was in the year of 1843 to 1910. He was a German general practitioner. He was the first to use solid media for culture of a bacteria using the agar as a solidifying agent, and he introduced the methods of isolation of bacteria in the pure culture, and he described the method of hanging drop technique which is used for detecting the motility of microorganisms, and he discovered some of the bacteria such as anthrax bacilli, tubercle bacilli and cholera bacilli. He introduced the staining techniques such as aniline dye. You introduce the staining methods by using the aniline dye. Then coming for Robert Koch phenomena and postulates. What do you mean by Koch phenomena? Koch phenomena is nothing but of hypersensitivity reaction when injected with tubercle bacilli or its protein seen in a guinea pig previously affected with tubercle bacillus. What it states means a person or animal already infected or previously affected with a Mycobacterium tuberculosis, they will show some hypersensitivity reaction when they are injected with tubercle bacilli or a protein. This is the statement. Then coming for the Koch postulates, here postulated criteria for a microorganism to be a causative agent of infectious diseases. Here there are some postulates, so we are going to learn about all the postulates. Coming for the Koch's postulates, here the first postulate is microorganism should be constantly associated with the lesion. What do you mean by lesion? Lesion is nothing but of wound. Okay, microorganism, they should constantly associated with some of the wound infections. Okay, this is the first postulate, and the second one is isolate the organism in pure culture from the lesions. You need to isolate. You need to pick up the organism from the pure culture from the lesion. Okay, the third postulate is same disease must result when isolated microorganism is inoculated into suitable laboratory animal. See, when you are going to inoculate the same microorganism to the any of the laboratory animal, the yield what you get from that laboratory animal, it should be the same. And the fourth postulate is should be possible to re-isolate the organism in pure culture from the lesion produced in the experimental animal. If you are taking any lesions from the experimental animal, you need to re-isolate the organism in the pure culture. Okay, that is the fourth postulate. Later, fifth criteria was introduced. What do you mean by fifth criteria? Means it states that antibody to the causative organism should be demonstrable in the patient serum. When you are going to take a patient serum, and if you are demonstrating or if you are diagnosing a particular antibody should be present in that patient serum. These are the exception postulates of Robert Koch. What are the exceptions to the Robert Koch postulates? Means Mycobacterium leprae and Treponema pallidum cannot grown in vitro, can be maintained in experimental animal. That was an exception. And Neisseria gonorrhea, no animal model. You said like no animal model, it can be grown in vitro. Then coming for molecular Koch postulate standing Falwok in the year 1988. Gene coding. Gene should satisfy all the criteria for Koch postulates. Virulence triad should be associated with pathogenic strains than the non-pathogenic strains. Virulence triad should be associated with the some of the pathogenic strains which are non-pathogenic strains. Okay, inactivation of a gene associated should subsequently decrease the pathogenicity. If the inactivation of a gene is associated with, subsequently you need to decrease the pathogenicity of the virus or a microorganism. Okay, then you need to replace the mutated gene with a normal wild type gene. Should be fully, it need to restore the pathogenicity, and gene should be expressed at some point during the infection and the disease process. Immune system or antibodies cells are direct against the gene products which should be protected from the host. The next contributor is Paul Ehrlich. He was born in the year of 1854 and he died in the year 1915. He was one of a German scientist. He is known as father of chemotherapy. He was the first one used to report the acid form nature of tubercle bacillus. As you all know, Mycobacterium tuberculosis has a acid content, that is a mycolic acid in the cell wall. He was the first scientist to observe that acid fastness of the bacteria, and he developed some stain techniques such as to stain tissues and the blood cells, and he proposed one phenomena that is called as Ehrlich phenomena. It is proposed at toxin-antitoxin interaction. That phenomena is regarding to toxin and the antitoxin interaction. Then he introduced the methods of standardizing toxin and antitoxins. Then he also proposed the side chain theory for antibody production, and he discovered salvarsan. It means it is one of a arsenical compound. It is called as magic bullet used for the treatment of syphilis, and he discovered a bacteria called as Ehrlichia, was named after him, Paul Ehrlich. Okay, in 1908 he awarded the Nobel Prize in the physiology or medicine for his contributions to the immunology, and he was the founder of first director of Paul Ehrlich Institute in Germany. Then, what are the other contributors? Hans Christian Grams. He developed the Gramston in the year 1884, and Charles Gramberland. He constructed a porcelain bacteria filters which lead to discovery of some viruses. And Ernest Ruska. He constructed first electron microscope. Alexander Fleming. He discovered the penicillin. Good pasture viral culture technique in chicken brew. Lady Mary Wrotel Montagu Variolation for immunizing against smallpox. And then L.A. McIntosh, phagocytosis and term phagocytes. M.E. Killenberger, L forms of bacteria. Barbara Emleclick, transponers. Walter Gilbert and Frederick Sanger, DNA sequencing, and Kerry B. Miller's discovered polymerase chain reaction and was awarded Nobel Prize in the year 1993. Then, some discovery of microorganisms, oxygen, Staphylococcus aureus, Neisseria gonorrhea, which is ban, Neisseria meningitis, Lophilus, Corynebacterium diphtheria, Frankel, Streptococcus pneumonia, Brucella melanitis, Kistesicu, Clostridium tetani, Hansen, Mycobacterium leprae, Yersin and Kitasketo, Yersinia peticis, and many more are discovered some of the microorganisms which you can go through these slides, or you want you can take down the screenshot of this slide. This is the same thing. If you want means you can take out the screenshot. Same thing. You can, if you want means you can take down the screenshot. For what thing they got Nobel prizes? Actually this is not for your examination purpose, for your extra knowledge. If you want means you can take down the screenshot of this. This is about introduction for the microbiology. Next, coming for bacterial taxonomy. Okay. Bacterial taxonomy. The first thing is classification. What do you mean by classification? Arrangement of bacteria into taxonomic groups on the basis of biochemical, physical, genetic and morphological properties. What do you mean by nomenclature then? Naming of taxa according to their characteristics. Okay, identification for identification of unknown taxon and to isolate and identify the causative agent of a disease. Then bacterial classification. Claver and Smith classification was one of the most accepted system of bacterial classification. Molecular classification, they have 6 kingdoms: bacteria, protozoan, chromastia, plantae, fungi and animalia. That you already know, you have learnt in your plus 2. These are the bacterial classifications. Just take a screenshot of this. But as of now we are going to just we are using here the Clavier and Smith classification. Okay, here we are using Clavier and Smith classification. Can you see? Make out this here. This is the classification. Then what is the principle you are used to classify the bacteria? Means there are mainly 3 approaches. One is phylogenetic, addition and the molecular. What do you mean by this? We shall see. Phylogenetic classification is nothing but of hierarchical classification representing a branching tree-like arrangement. Can you see here like a branching tree-like arrangement? We differentiate it into bacteria, archaea and eubacteria. And we are given some examples of bacteria and for examples of archaea and some few examples of eukaryote. Okay, then coming for phylogenetic classification, characteristics which are given importance or morphology of bacteria, whether it is a cocci or bacilli, by staining property, whether it is gram positive or gram negative, by the cultural characteristics such as whether it is lactose fermenting or non-lactose fermenting colonies, in biochemical reaction whether they are coagulase positive or coagulase negative, based on the antigenic structure such as antigenic structure of somatic antigen present in the cell wall. For sexual sex antigen, they won't be present on the cell wall. Then the last thing is adenosine classification, phenetic classification, we call it in another one name, classifies organism based on giving equal weight to the every character. Okay, it giving numeric taxonomic system using an algorithm like cluster analysis. By the number representation, we are going to classify this. Then the last thing is molecular classification based on the degree of genetic relatedness of the organism, that you already know that base pairs. Okay, guanine and cytosine content of a bacteria is estimated from extracting DNA from a pure bacterial culture. Coming for the nomenclature, it is a branch of taxonomy that is concerned with designating scientific names for that particular taxa. Okay, the bacterial nomenclature is based on binomial nomenclature. Bi means nothing but two, based on two nomenclature as proposed by Swedish botanist, that is Corliss Linnaeus. Binomial nomenclature, what do you mean by this? Means scientific names for taxonomic levels above genus always capitalized but not italicized. Okay, for example, phylum Proteobacteria. Uh, a scientific name comprises of genus name, start with the capital letter and the species name. If you are writing a genus name, that should be start with a capital letter and the species name should be in the small letter. Both genus and the species should be written in the italic. Okay, and they need to underline. For example, if you are writing in italics, no need to underline. Okay, here I am given an example of Staphylococcus aureus. Okay, you know the example here, Staphylococcus aureus. It is in the italics. The genus name will start with capital and the species name will start with small. Or else Staphylococcus aureus, you can write it and you can make it underline. Genus will be usually Latin noun. Okay, species refer to the defined taxon of organism with a particular genus. The genus and the species are coined based on some property of bacteria such as Staphylococcus aureus. Staphyla means bunch of grapes. Aureus means golden yellow. So it will be like a bunch of grape appearance and it will shows yellow color on nutrient agar. Okay. Neisseria meningitidis discovered Neisseria. He discovered that, so we are given Neisseria and the disease causing is meningitidis. Brucella suis and Brucella melanotis discovered David Bruce and the animal host pig, and the place where discovered, that is Malta or Europe. So because of that we are given that name. Here are some of the example of that particular bacteria and the new name of that bacteria. If you wanted this, means you can take down the screenshot of this. Then International Reference Laboratory, which are designed as a type culture reference centers, that is ATCC and NCTC, that is American Type Culture Collection, which is in the USA, and National Collection of Type Cultures, it is in the UK. This is about the introduction and the taxonomy of bacteria. So if you are fine with this method, you can comment down you are very fine with this. If you want any extra things, so you can text me or you can comment me, or we have Instagram and YouTube, both the things, so you can text me in the particular like Instagram, or you can text on the comment box. Okay, thank you for watching, and before that please subscribe, and if you like the video, please like the video, and thank you.